lot of people will slowly but surely get in. Well, good morning, everybody. My name is Alessio Fasano. My role here today is as the coordinator of the GEMMA project that has been an unbelievable journey, 7 years of history that you're going to hear today with some, highlights and some preview of the outcome of a very impactful project related to autism. We are in this beautiful, venue. There is Salone dei Marmi of the town hall in Salerno. And it was only obvious and normal that we'll have the guy that is on this place in terms of managing to give us a welcome message. So Prefetto Panico is the Commissario Straordinario of the municipality of Salerno, and we ask him to eventually give us a few words of welcome.
Grazie professor Fasano, sono davvero lieto a nome del Comune di Salerno che mi onoro di rappresentare, di dare insieme al professor Alessio Fasano il benvenuto ai tanti ospiti europei, d'oltreoceano e del nostro paese a questo prestigioso congresso internazionale. E lo faccio in questo Salone dei Marmi, dove è passata la storia. Dopo lo sbarco del 9 settembre del 1943, con la svolta di Salerno, proprio qui, il 22 aprile del 1944, c'è stato il primo governo politico post-fascista. Ed è uno scenario questo adeguato all'importanza di un evento che segna, caro professore, dopo un lungo percorso, un percorso di 7 anni, il momento cruciale di un progetto scientifico e tecnologico, ho letto qualcosa, ma di altissimo profilo e al tempo stesso, questo mi piace sottolineare, di grandissimo, profondo significato umano, dedicato com'è al primo studio prospettico al mondo. E questa è una cosa che mi fa molto onore, che monitora bambini a rischio autistico fin dalla nascita. E soprattutto è presentato come un approccio innovativo, in grado di aprire nuovi orizzonti alla medicina, sia preventiva e sia di precisione. E io vorrei dilungarmi tanto, essendo un profano però la voglio smettere subito, perché ad illustrare i dati raccolti ed elaborati, i risultati preliminari con le ricadute applicative e con le prospettive di applicazione scientifica, e saranno il relatore di fama del mondo accademico e scientifico, ci sono operatori sanitari, professionisti di fama, di grande fama, ed è lungi da me di sottrarre ulteriore tempo prezioso ai lavori. E quindi chiudo subito augurando a questo congresso il grande successo che merita. Auguri.
Good morning, everyone. Thank you. Let me say thank you to the organizer and in particular to Alessio Fasano for inviting me, to give me this opportunity to moderate this final conference of the GEMMA projects. I am a science journalist. I'm not a physician. I'm not a researcher anymore. So the first talk of this morning will be about autism and information. And if I can have the first slide, Questo? Freccia in giù. Okay. No. Yes. So, today, with my talk, I would like to dive into a topic that is as much as about science as it is about human rights. Autism information in an era like this one, called infodemic era, where misinformation spread faster than facts, distinguish between myths and evidence-based medicine is not just an academic exercise. It is a necessity for millions of neurodivergent individuals. So just when we talk about fake news and misinformation, I would like to give you just an example of what your patients and children, and most parents are exposed to. So just a short, no. Can I start the video?
Decided, let's do just a study that just isolates the MMR vaccine. So they did a study of children in Georgia. They're senior scientists. And his data come back, and they show that with certain groups of children, with black children, for example, and they didn't look at people who got the vaccine and people who didn't. They looked at people who got the vaccine, children who got the vaccine during the first 36 months of life, and people who got it later or not at all. Those were the two groups. And what they found is that children who got the vaccine on time, which was before their third birthday, had a 350% greater chance of getting an autism diagnosis than kids who did not.
So, as you can see, this is just an example of dangerous fake news about vaccine and autism. But the real question, So the real question is why autism is such a magnet for fake news compared to other fields of medicine. First of all, this is a complex disease, is a complex condition. And when science has not an on-off switch for autism and offer nuances, or unfortunately we don't know yet answer, misinformation offers certainties. So the first element is that is a complex pathology, is a complex condition. And the second one is that family often feel overwhelmed when they receive the diagnosis of autism, making them vulnerable to charismatic figure promising miracle solution. And the third one is the social era. Of course, social media algorithms often prioritize engagement over accuracy, and creating a sort of perfect storm for misinformation around autism. But we cannot talk about misinformation, fake news, and autism without mentioning the, the 1998 Wakefield fraud. It is the patient zero of medical misinformation. Based on just 12 children and falsified data, it claimed a link between the MMR vaccine and autism, the same vaccine that Mr. Kennedy quoted in the video we just saw. Even though the study was retracted by Lancet and the author disgraced, the myths persist today. Let's be clear, it's just a message for also Mr. Kennedy. Decades of global research involving millions of children have proven beyond any doubt that vaccines do not cause autism. But beyond vaccine, we face the miracle cure industry. This is where misinformation become physically dangerous, really dangerous. Some group promotes MMS, which is essentially industrial bleach, to cure autism. I mean, in Italian is candeggina to cure autism. Others suggest chelation therapy to remove non-existent heavy metals, which has led to recorded deaths. The problem is that they are not just alternative options or alternative therapies. They are harmful practices that exploit hope and cause real trauma. So what is the scientific truth? We will see a lot of data this morning, and we will discuss in a media conference in the afternoon, but as far as we know now, science tells that autism is a neurodevelopmental condition with a strong biological and genetic basis. It is about the brain, how the brain is wired. It's heritable, of course, involves a complex interplay of hundreds of genes, and is not something you catch, nor is caused by bad parenting or modern lifestyles. Modern science is moving away from the idea of fixing a broken person. We are embracing the concept of neurodiversity. This shift views autism not as a tragedy to be cured, but as a different operating system. Our goal should not be to make an autistic person appear normal, but to provide the tools and environment they need to train. So, how do we fight back? This is just a slide I would like to show you. I think this is very important for physicians, for psychiatric and physicians in general. It's important to give to families, to parents, some red flags. This is just an example. But when they meet this kind of words or wording on the social media, on the web in general, promise of a cure, secret of hidden knowledge where big pharma don't tell you, testimonials and anecdotes, they use often testimonials as source. They use a video on YouTube as a source, a scientific source. Or high cost for exclusive treatment, you have to be very careful. This is an important message. I think this is just an example, and I think that we can find a lot of more. We, of course, I am a journalist, and I am aware about the responsibility as my category has about the spread of myths or fake news or whatever. The first thing I would like to stress, and we will discuss in the afternoon, is about avoid the false balance, giving a scientist and conspiracy theory the same airtime is not neutrality, it is dangerous. But also, I think professionals, healthcare professional, and all the other stakeholders, we can start together and try to do a better job in the future. This is my conclusion. I think that today's conference is called the final conference, but in another point of view, in my point of view, it's just the beginning. And then I think that starting from the GEMMA results, we can altogether, media experts, journalists, communication, researchers, physicians, and all the people involved in this project start a discussion and start a project to disseminate the real science data, not in order to debunk what the fake news says, but in order to do a great job of pre-bank. Thank you very much. So, I invite the first, I don't remember the name, the first speaker of today is Alessio Fasano. Amnesia. Okay. So here is to you.
All right. Good morning again, everybody. And as Massimo said, you know, when we were entrusted by the European Commission to do this study was a major undertaking because the confusion, the myth, the misunderstanding, and the complexity of a condition that, by the way, is not homogeneous. It's called spectrum for a reason, because the final destination is the same, but how these kids, they got there. is definitely very different. So it's not going to be a magic bullet that will fit everybody. The other thing that, prompted us to do that was the social aspect of the story. As Massimo said, with the prevalence in only 40 years, moved from 1 in 10,000 to 1 in 36 in the United States, 1 in 42 in Europe, it's pretty obvious that we're talking about something that touched every single family. Somebody in the family will have it. And I want to start with the final fact. This was a project of this magnitude that, again, as Massimo said, was not aimed to fix this. We are not promised a cure. But the foundation to have a better understanding, to move forward, to really put factual evidence and a very intricate, biological question. Why this wiring that leads to autism is increasing over time so exponentially? All chronic inflammatory diseases are in a rampage if we embrace a Western lifestyle. But the acceleration we've seen this is unprecedented. And to tackle something so complicated, it would take a dream team. And this was indeed a dream team, 16 partners, both public and private, from both the sides of the ocean. So we have 2 entities from the United States, Harvard Medical School and Johns Hopkins University, and many very top schools in Australia, in Europe, that came together, and companies that came together to solve the problem of this, situation. The title and the name of the project is GEMMA. It is the acronym of what is the essential elements that we thought, and you will see in my presentation the rationale why we went this way, that is needed to create this foundation to build the house, a better understanding of autism spectrum disorders. GEMMA stands for genome, so genetic component is fundamentally important. Environment. You can't blame only genetics when you have an acceleration in only 30, 40 years of the same generation on genetic mutation. So the environment plays a role. Microbiome, and this was a call from the European Commission, very visionary, focused on microbiome. Metabolomic, because the crosstalk between our genes and the microbes' genes will activate specific metabolic pathways that pathogenically, at least this was the ambition that we have, and we hear about that, can eventually lead to the outcome. That's the reason why the acronym. So we got a good budget to work with, 12.4 million euros. I told you the 16 partners. This took 7 years because, to just increase the challenge of this study, in the midst of this study, where we already start to recruit patients, when already our colleagues start to do the animal models, an unforeseen, never experienced by this generation before, a pandemic hit us and everything was shut down. So we had to sacrifice all the animals. We had to stop recruitment. The diagnosis of autism is based on in-person interview that was not available anymore, and we have to reinvent and repurpose the project to make sure that we'll continue. So this team has shown resiliency. These 100+ people that work on this project really came to rescue because they were not doing this for anything else but as a service to increase the knowledge and give those foundations I was telling you before. Again, to give you a sense of the magnitude, more than 1,400 people in these 7 years have been recruited, more than 23 biospecimens, blood, saliva, urine, stools have been collected, trafficking all over the world to go to labs to be analyzed, maintained in a biobank, is the richest biobank that we have right now, and more than 720,000 metadata. This is astonishing wealth of information that's been generated at the service of the scientific community. What you're going to hear today is the first scratch of what is going to be years to come of outcomes of this foundational stuff that is being made. All this will be, and will be, as you will hear today, analyzed also by AI modeling To do what? To do some prediction of why some kids started with a genetic predisposition. Over time, they take the wrong turn and will develop again ASD and others that will keep straight and will not. So why we want to do this? Again, as I told you, this was one call that was specifically focused on microbiome, and I believe at that time was a strong supposition, now is a reality. The autism is probably the quintessential example of miscommunication between the gut and the brain, what we call now gut-brain axis. This is not disputed anymore. We know that the brain communicates with the guts. We knew this forever because when we got nervous, the first thing that we somatize, it's with the GI symptoms. A relatively new notion is that the gut can communicate with the brain. And then there is this problem. The word the microbiome was on a halt. I mean, probably the microbiome literature has been the richest of the history of biology. We have now an average of 2,000, 2,500 papers a month published on the microbiome. Unfortunately, the vast majority of these studies are descriptive. They are not mechanistical enough to translate that information to clinical implementation. And therefore, the entire world of the microbiome was at this crossroad. They said, how are we going to move from there? And, you know, this shift from descriptive mechanistic, again, is fundamentally important to capitalize on the microbiome, but also omics, to make use of that for two reasons. One, biomarkers for patient stratification that will be fundamentally important for autism, because I told you this is not an homogeneous situation, and most importantly, possible point of attack for treatment. And, you know, there was also another problem that we were facing, lack of standardization. So, you know, how you standardize the work on microbiome, how you collect the samples, how you ship the samples, how do you extract the DNA, how do you make libraries, what kind of platform you use, the taxonomic assignment, what kind of reference point do you have to have. That was also a work in progress at that time. So, and the project contributed also to make the rule of the game, so to speak. But the key element that made this study one of a kind, as you heard from the prefect in his introduction, is the longitudinal nature. Cross-sectional are already challenging clinical study. Prospective to follow over time and for 7 years in a population like this, it is extraordinary. I, as a father and mother with the kids with autism, know that that's a full-time job. My life is changed to care for that child. Now there's another baby coming in, and I'm told I need to be in touch with you every 3 months. You need to answer a questionnaire. Every 6 months you need to give me samples, and then I'll come back if you don't do it, and this for 7 years. So that was very, very challenging. And, you know, again, this explosion of work on the microbiome is also reflected in the microbiome in autism with the same kind of situation. And there are, you know, very limited evidence. Again, fake news. You may see the error that they say, oh, the microbiome has changed and therefore this is the reason why they develop autism. Not necessary, so it can be the consequence and not the cause. And only the prospective study and focus early life, because if you will see this today, if you do the text too late, you miss that window opportunity to understand what's going on. And then, you know, even systematic reviews said, you know, guys, let's take a look what has been done so far. And the answer is not enough. We need more longitudinal studies. Again, this is bidirectional. And there are 3 avenues that the gut communicates with the brain. The first one is the nervous pathway. So the highway that is used is the sympathetic and parasympathetic nerves, the vagus being the most important, that you will hear how important is the balance between the two. The second, very fashionable nowadays, the endocrine, you know, pathway, i.e. the fact that the gut can produce hormones that can change our behavior. Classical example, GLP-1, that change our behavior in terms of feeding, i.e. appetite. And then the third one, the intestinal barrier permeability with the antigen trafficking, inflammation that can spill into the brain and therefore create change in behavior like the eating behavior, as I told you, but also cognition, impulsivity, anxiety, depression, and so on and so forth. Very active field of research, extremely active. This was the premise. So how we designed this study. The premise was, okay, the gut-brain axis is conditional to healthy gut and healthy brain. If the gut is healthy, so the barrier works well, there is compartmentalization, there is a good microbiome symbiotic relationship with the host, antigen trafficking is tightly controlled, the immune system is doing nothing, so in other words, you have an energy at the mucosal level, the brain is healthy. Conversely, something goes wrong in the gut. Permeability increases, so antigen traffic increasing. The immune system now is bombarded by stimuli to do inflammation. And the microbiome changed to the point in which you have this dysbiosis that leads to the point in which we have some microbes that can not only create inflammation, but be improved, as you will hear also from the studies that Aletta will present, change in behavior to the point that they've been labeled as psychobiotics. Now you have an unhealthy gut and you have an unhealthy brain, or dysfunctional brain, or inappropriately wired brain, whatever you want to call it, because that's the other thing. Who said what is normal here? Because this is a spectrum. You know, I believe to be in the ADHD spectrum personally. So what is the threshold? So, but you know, all this to say, there is a lot of discussion here. One thing that we didn't know well was one of these elements of permeability, how that happened and how it's changed. That was our contribution almost 25 years ago when we discovered this molecule that now is used as a biomarker of gut permeability. This molecule is called zonulin. It's been object of more than 1,300 papers. Anything that you can imagine that has to do with chronic inflammation has been linked to this molecule. In red, you see those that has to do with the gut-brain axis miscommunication. So there is a lot of work here, and this leads to the concept of what everybody's focused on, what we call gut health. Gut health doesn't mean only GI issues, it means our health. And gut-brain axis miscommunication, autoimmune spectrum disorders is one example of what can go wrong when these three pillars that dictate gut health, the immune response, the microbiome, the gut permeability, they don't work in synchrony. And these three pillars, they influence each other, by the way, because if you have dysbiosis, you have elevated production of zonulin. If you have too much permeability, that goes in dysbiosis. Of course, increased permeability, antigen trafficking, there is an immune system hyperbiligen, and so on and so forth. And you will deal with all this, including this gut-brain connection. Now, to exemplify why we did GEMMA and the rationale why, I take a one-moment digression and show you an animal model that has these three pillars affected. So, in other words, gut health is jeopardized. This is a genetically engineered mouse that produced too much zonulin. So this is a zonulin transgenic mouse. The structure of the intestine is normal, but the permeability, of course, of this mouse, of the intestine, is elevated. So there is loss of control of antigen trafficking. Consequently, this mice microbiome completely different from a wild-type microbiome. And if you ask what is the difference there that makes this difference, they lose, and you will hear this as a product of GEMMA, protective elements, like, Akkermansia, and you see another representation of pro-inflammatory LPS-loaded microorganisms like Rickenella. That's make the difference. The immune system of these mice also is different. Again, I can't go into details, I don't have the time, but the immune cells, they are supposed to put the brake on inflammation in this mouse are underrepresented, and the immune cells that should induce inflammation, that induce inflammation are overrepresented. The other interesting thing of this mouse is hyperactive. So this is a very simple, test in which you have a camera here, you have the mouse here, and you trace through this camera how this mouse moves in the cage. A typical mouse spends a good chunk of time at the edge of the cage because the center it's too risky because predators can be there. And this is the zone transgenic mouse. You see red everywhere, many more lines. So it's hyperactive and doesn't have a purpose. It moves randomly. And if you look at the brain of these animals and look at specific, expression of genes, what you find is that some genes that can control the blood-brain barrier are, expressed in a way that the blood-brain barrier leaks. So not only the gut leaks, but the blood-brain barrier leaks. And also genes that control inflammation are upregulated. So in other words, there is neuroinflammation in this animal. So far, so good. But what is interesting, if you take out of these animals the microbiome component, so you make these animals treated with a cocktail of antibiotics, so there is no microbiome there. Oops, what happened there? You revert the phenotype, so it behaves like the wild type, and also the gene expression will revert to normal. Meaning, in other examples of this communication between the gut and the brain that leads to phenomenal, outcome. Based on all this, we, put forward this hypothesis that this is the march from here to here, from health to disease. This is a gut barrier. It's normal at the beginning with the, symbiotic relationship. So everything works well. The metabolome is in balance. The antigen traffic is tightly controlled. Then you have this bridge to the barrier that moves here. Antigen traffic increases. There is an innate immune response that eventually creates inflammation mediated by pro-inflammatory cytokines that per se can increase gut permeability. And now you're in this vicious loop and you break, the tolerance, and then you develop, the neuroinflammation process. And again, all this, with some, modeling that we predicted to be there, and we can use here from Thanos, and then, eventually microbiome and metatranscriptomic plus metabolomics that we're going to hear from Jacopo and Corrado. So this was our premise. And as, you heard, many times, it's all matter of connections. In other words, the synaptogenesis and myelinization of these kids is not the one that is predictable in a situation of physiological maturation. And also the pruning, once the synapses are made, because, that's another thing that is interesting, that the brain during development will make more synapses that we need, and that are pruned based on the experience and stimuli that these kids will have until we get to the proper number. And that these two processes seems to be not appropriate. I told you already this amazing increase in prevalence over time, and give me another message. We are not born to develop autism. We're born with a predisposition. And this predisposition will translate in clinical outcome, depending on how we play our genetic cards. The challenge, GEMMA, was to understand what we do to play these genetic cards in the wrong way and what we can do to change the game so that we play this in a more productive way. This was the modeling that we put together to identify specific biomarkers in yellow. And the modeling, the predictive modeling, will be discussed by Thanos and Francois. Francois will not be with us, so Thanos will present this. But these gentlemen are the ones that really were focused to predict what could be seen in GEMMA. Then we had a phase that was the preclinical phase, similar to the experiment I showed in the transgenic mouse. Here, on the other hand, we use a mouse model that are humanized with stools coming from kids with autism, and Aletta is going to tell us about it. Then we had two clinical arms, an observational arm in which we just follow these kids at risk. These are the inclusion criteria was these kids needs to be born by a family in which one brother or sister needs to have the problem, and Dario will talk about that. And then the most exciting part of the story, those that eventually will develop the problem based on all these omics and based on those predictions, what Aletta found in the animal model, can we have an interventional arm? Something that will allow us to manipulate, one of the three pillars of gut health, specifically the microbiome, to see if this will change something. And Geraldine, that was in charge of most of this trial, would tell us about this. the understanding, of course, this is a proof of concept, open label, no control, no nothing, but just proof of concept, okay? So we are not offering cure here. I want to just make sure. And then finally, this modeling with artificial intelligence and multi-omic analysis to identify potential biomarkers to build predictive model. And Corrado and Jacopo will wrap it up and will tell us about this. What you're going to hear is this. So I will give you a snap preview. What we learn the march starts probably, and I want to stress probably, with an imbalance between the sympathetic and parasympathetic system that in turn cause this biosis that we can see even a year before the onset of the disease. Then there are some epigenetic immune epithelium reprogramming 9 months before. This breach of the barrier zone independent will happen 6-9 months before. Then there is the activation of pro-inflammatory response, and that will lead to the onset. 4 points of potential attack to stop that, march. So that's synthesized what you're going to hear in the next few hours. Again, it took a village. It was an enormous amount of work. Far to be finished. As Massimo said, this is not the end. It's the end of the project, but the beginning of a new era in the world of autism. Thank you.
Thank you, Alessio, for your amazing overview of the project. And now we go with the second session, and we see all the aspects. Start with Aletta. Aletta Kranevelde from Vrije University of Amsterdam. She will talk about what we have learned from animal models. Thanks.
Thank you very much for your kind introduction, and I'm very pleased that I can stand here on behalf of a whole team who worked on the preclinical studies. And I also stand here with a heavy heart that we now are about to wrap up this beautiful, really adventurous journey we had in relation to studying the gut-brain axis in autism. Today I will present what we have learned from all our animal work, and this was done in collaboration with Danone Nutritia Research from Utrecht in the Netherlands, but also a large part was performed at INRAE Michaelis in France, close to Paris, and at Utrecht University in the Netherlands. So the objectives of the preclinical trials were to explore whether we could transfer the behavioral phenotype of autism via human fecal transplantation into mice. The next objective was to explore whether the autistic phenotype in mice is dependent on microbiome-induced immune changes. And last but not least, we performed some dietary interventional studies mimicking the dietary interventions performed in the clinical trial just presented by Alessio to target the microbiome dysbiosis in mouse models. So the core of this project was working with stool samples of kids diagnosed with autism and their siblings. So we are very, very grateful for ASL Salerno for recruiting the families, which resulted in 16 samples we performed the studies with, 4 samples of children diagnosed with autism and their sibling controls, and 4 samples of kids diagnosed with autism and suffering from GI problems and their sibling controls. So before we started doing the human fecal transplantation studies at Danone Nutritia Research, they investigated the composition of the samples we obtained from Italy. And they looked with 16S RNA sequencing at the bacterial composition and found that the composition of those kids diagnosed with autism was closer to the siblings than those diagnosed with autism with GI problems, intestinal problems, which is shown here on this, oh, you can't see the pointer, on the left-hand side of the slide. In addition to that, they looked at the production of short-chain fatty acids in these samples, and they found no differences in the production of acetate and butyrate. in all samples from the kids, but they found that there was a higher production of propionate in those samples from kids diagnosed with autism. In addition to that, they did some fermentation studies using galacto and fructo oligosaccharides. These are non-digestible oligosaccharides which induce the production of beneficial short-chain fatty acids, galacto and fructo oligosaccharides, and they noticed that the combination of these two induced the most short-chain fatty acids after 56 hours. And in addition to that, the combination of galacto and fructo oligosaccharides induced the abundance of more beneficial bacteria, the Bacteroides and Bifidotaxa. So we used for the studies at INRAE and at Utrecht universities only the samples of the kids diagnosed with autism and suffering from intestinal problems and their sibling controls, because we found a difference in the composition of the bacteria in these samples. So we transferred these into our mouse models. So this is giving, sorry for the busy slide, but this is giving an overview of our major findings. So in France, they studied the transplantation of stool samples into germ-free mice. In Utrecht, we studied the transplantation of human stool samples in microbiome-depleted animals, and we did that by lavaging the intestinal tract to clean off the bacteria in there. In addition to that, at both sites, at both institutes, we tested two different mouse strains. The bulb C mouse, which is more a Th2 responding mouse, which means it's more prone to develop allergic reactions and it's more prone to be protected against parasite. And the black 6 mouse, which is more a T helper 1 responder, which means it has an extensive cell immunity and it's very much protected against parasites. And is prone to develop more autoimmune-like problems. When we transplanted the stool samples of the kids into these two mouse strains, we saw that there was in both in the germ-free mice as well as in the microbiome-depleted mice, there was a distinct profiles of the microbiota. Not only the composition, but also the activity, so the metabolites produced in these mice. And the striking thing is that in both models, the germ-free as well as in the microbiome-depleted animals, we could transplant with the stool samples and create an autistic-like behavioral phenotype. In the germ-free animals, this was in the black 6 mice, where we showed repetitive behavior and a compromised spatial memory. And in the microbiome-depleted animals, we noticed more exploratory behavior and impaired social interaction. In both models, we saw a disbalance of the immune regulation, and we measured that in spleens of these mice. And in the microbiome-depleted animals, we saw the induction of neuroinflammation in the prefrontal cortex, and we saw that also there was a leaky gut in these mice, which is an impaired intestinal barrier, where we saw also a decreased mucosal immune response, and we measured that with the measuring soluble immunoglobulin A. In the germ-free animals, which were transplanted with the stools of autistic kids suffering from GI problems, we saw that the propionate-producing genera correlated positively with the impaired spatial memory. The studies, one is published already, and the other one is recently submitted, and both hopefully will be ending up in the nice journal Gut Microbes, and was part of the work of 3 PhD students, Lea Rossini from INRAE, and Naika Prance and Lucia Peralta from the Utrecht University, who all defended their thesis during the work during this past 7 years. And I should mention also that Tampere University played an important role in the analysis of the data, and we are very grateful for them. So we performed more studies, in this case in Utrecht, where we transplanted human fecal matter from ASD kids and their sibling controls in a mouse model, which is called the BTBR mouse, which is more or less a genetic model often used in autistic research. And we transplanted these mice with the stools, and what we noticed is that those BTBR mice, which were transplanted with fecal samples of kids diagnosed with autism and GI problems, they showed a more enhanced repetitive behavior than the ones not transplanted or transplanted with the sibling controls, and this more enhanced repetitive behavior was associated with neuroinflammation in the brain, a systemic immune dysregulation, and also an impaired gut barrier function. And also what we noticed investigating the intestinal microbiota and all these 4 groups is that they have significant different clusters with associated metabolic pathways. So another study is performed as well in Utrecht, but always in close collaboration with INRAE. In previous studies, we showed that in a food allergic model, mice show autistic-like behavior. In this case, we used cow milk's allergy, and we transplanted mice which were depleted, obviously, from the microbiome with the stool samples of autistic kids and their sibling controls, and we look at the cow milk's allergic response, and we saw that if these mice were exposed to human microbes from ASD, that they exhibited a more pronounced impaired social interaction compared to the ones transplanted with sibling stools. And this was associated with astrocyte activation in the prefrontal cortex in the allergic mice. The transplantation of the stools of kids with autism into the mice did not as such affect the cow milk allergic response we found in these mice, but we saw a reduction of antigen-specific immunoglobulin G2A. What did happen is that those mice which were transplanted with ASD stools, they showed a reduced mucosal cow milk allergic reaction, and that was associated with a reduction of soluble IgA. And again, transplanting these mice with all these different stool samples from sibling controls and kids with autism and GI problems, that there was a significant different clustering of the composition of the microbiome, as well as induced by the food allergic response as well. So the next step was to explore whether the autistic phenotype is dependent on microbiome-induced immune changes. And this is a pretty complicated study, and I hope I can lead you through. So please remember that we investigated the effect of the transplantation of stools from autistic kids and looking at the cow milk's allergy response in these mice. And we used these mice as donor mice for our next study. So it is explained here on the left-hand side. So we had 4 donors, non-treated animals, which were the controls of the allergic mice. So we had control allergic mice. They didn't receive any transplantation. And we had allergic mice which were transplanted with the stools of siblings or autistic kids. And we took the spleen cells out of these mice and transferred them to naive donor mice. And then we explore several things. And the first highlight of this study is that if we transplant the splenocytes of these 4 different donor mice into recipient mice, we noticed that the splenocyte transfer had a huge effect on the composition of the microbiota and the activity in the GI tract. So we saw clear clusters, which is depicted on the little figure in the middle, where we can see that they really differ, the control allergic, sibling allergic, and ASD allergic splenocytes. And that was also found at a family level, where we noticed that the splenocytes obtained from animals which were exposed to ASD stools and went through an allergic response, that they showed an increase in the amount of bacterial taxa compared to the sibling allergic group. So this was really a striking result, showing that the immune cells indeed can directly influence the composition of the bacteria in the gastrointestinal tract. So the next step was to study whether it could also transfer the behavioral phenotype found in the cow milk allergic mice. And what we noticed is if you transfer splenocytes from allergic animals, donor animals, they didn't receive any transplantation, and you put that in naive recipient mice, you could transfer the autistic-like social avoidance behavior after oral challenge of these naive animals. So you can transfer behavioral changes by transferring and inducing an allergic response in recipient mice. The striking thing was, if these splenocytes were obtained from animals either exposed to sibling or stools of autistic kids, then it got completely mixed up. So what we saw is that if there was an exposure to human microbes in the donor animals, that didn't result in autistic-like behavior in the recipient mice after challenge. And that was also associated with a reduced neuroinflammation in the prefrontal cortex and also in relation to a diminished allergic response either in the gastrointestinal tract but also systemically. So basically human microbes really sort of hijack the allergic function of splenocytes. So the last bit of my presentation will be about the dietary intervention studies we performed in Utrecht, but as well as in INRAE, in our mouse models. And first, I would like to show you a study we performed in Utrecht in close collaboration with Danone Nutritia Research, where we did a prebiotic intervention in a new mouse model for autism. In this case, we expose mice in utero to valproic acid, and it is noticed that the male offspring of these mice show autistic-like behavior and intestinal phenotype. And valproic acid is also related for pregnant women to have a higher risk to give birth to children who will get a diagnosis of autism. Anyway, so we used this mouse model, we worked with it a lot. It's originally developed by Jane Foster from the States. And what we did is, when the animals were born, we started to feed the mums with a dietary intervention containing of the galacto and the fructo oligosaccharides, the non-digestible oligosaccharides, which are also referred to as prebiotics. After weaning of the animals, the male animals were kept on the same diet, a control diet obviously, and a prebiotic diet. And we noticed that if you feed these animals from the day they are born through the mother or later after weaning eating themselves, is that galacto and fructo oligosaccharide combination rescued the impaired social behavior found in these mice, but also the cognitive problems. And that was associated with a reduced loss of serotonergic neurons in the rapha nuclei in the brain, and also reduced neuroinflammation in the cerebellum. It reversed the pro-inflammatory T-cell response in the spleen, so it corrected the disbalance of the systemic immune system, and it also restored the intestinal barrier function. And what we also noticed is that this intervention restored the key intestinal taxa, which was downregulated in these in utero exposed, VPA exposed mice. So this is only the prebiotic fibers. So when Danone Nutritia entered the GEMMA program, they were the ones who were in charge to develop a synbiotic dietary intervention. And synbiotic means a combination of a probiotic strain with prebiotic fibers. And based on the studies we performed, they chose for the prebiotic fibers galacto and fructo oligosaccharides 9:1. And they identify Lactobacillus paracasei as the preferred probiotic strain to use in the clinical intervention studies, and also we used this intervention in our models. So they first demonstrated that all the Lactobacillus strains they identified did grow very well on the prebiotic fibers, which is known to be inducing beneficial bacteria, so that's on the left-hand side. In the middle you see that exposing the stool samples of autistic kids to this symbiotic intervention and culture them for 56 hours, that the combination of Lactobacillus paracasei and galacto and fructooligosaccharides increased the bifidobacteria and decreased the pro-inflammatory Peptostreptococci bacteria. And also they noticed that the whole soup of microbes was becoming a saccharolytic community, which means that they really digested the prebiotic fibers and increasing the amount of short-chain fatty acids. So in Utrecht, we performed an intervention study with the symbiotic diet where we combined Lactobacillus paracasei with the prebiotic fibers, but we also choose to use a dietary intervention VivoMix, and VivoMix is in a clinical trial demonstrated to be beneficial for kids diagnosed with autism and suffering from GI problems. And this VivoMix contains of 3 lacto strains, bifido strains, and we also did the combination of VivoMix with Gosfos. So that is diet A and B, and diet C was the combination of Lactobacillus paracasei with the prebiotic fibers. So what we did is we fed animals transplanted with stool samples of kids diagnosed with autism and GI problems and their sibling controls to these 3 diets and obviously a control diet. And what we found is that there was a distinct effect on the distinct differences of the profiles of the microbiota measured in stool samples of these mice, the composition and the metabolism induced by the diet, where diet B and C containing the prebiotic fibers was more pronounced in its effect than diet A. Diet C with the Lactobacillus paracasei and Gosfos was improving intestinal barrier better than diet B and diet A. All diets had an impact on the systemic immune disbalance in the ASD transplanted BTBR mice. Diet A reduced neuroinflammatory response in the prefrontal cortex of the BTBR mice transplanted with ASD stools. And unfortunately, we didn't measure any effect on the behavioral phenotype in this mouse. At least not on the things we measure, and this in this case was measured, was the repetitive behavior, the major behavioral outcome. So the diets didn't have or had very little effect on the behavioral output of these mice. In France, at INRAE, they tested the same symbiotic intervention, which was called diet C in the previous slide, so the combination of Lactobacillus paracasei and the galacto and the fructooligosaccharides, and they studied this in germ-free bulb C mice, which were transplanted with the stool of kids with autism and GI problems and their sibling controls. And what they found was, again, a distinct microbiota profile, the composition and the metabolism in ASD and sibling receiving mice with the control diet, but also with the symbiotic diet. And they also reproduced the behavioral impairments, stereotype behavior and impaired spatial memory. As in the first study, they noticed that the dopaminergic neuron system was modified in the ASD transplanted mice. And the diet, diet C, the symbiotic diet, they modified the gut microbiota composition and metabolism, but had limited impact on the behavioral alterations in the ASD transplanted mice. So this brings me to what did we learn from all our preclinical work. So we learned that intestinal microbes indeed are regulating behavior in mice. I didn't present results here, but this was a beautiful study performed at INRAE, where they looked at germ-free animals and conventionally colonized animals. And there is a role for the serotonergic system in that. We showed that human microbes from kids diagnosed with autism and suffering from intestinal problems can induce autistic-like behavior, but also an intestinal immune and neuroinflammatory phenotype in mice. We showed that splenocytes transfer affects the composition and the activity of intestinal microbes in recipient mice. We also showed that we can transfer with allergic splenocytes autistic-like intestinal and behavioral phenotype in recipient mice associated with a food allergic response. And we also showed that human microbes hijack allergic splenocyte transfer associated immune and ASD-like behavioral phenotype in recipient mice. Another part of this program was performed by a PhD student, Huangleng Zheng, in Utrecht. I didn't show the results because due of the lack of time, but we noticed that ASD-associated bacterial metabolites, p-cresol, but also 4-EPS, is affecting the microglial function. The microglial are sort of immune cells in the brain, and they affect them via effects on SHADases, and SHADases are important enzymes which regulate synapse function, so the connection of neurons. And we showed that indeed that these SHADases are upregulated, and if you inhibit that in an animal model, you see, for autism, you see it normalizes behavior, but demonstrating that indeed bacterial metabolites which can reach the brain can also have direct effects in the brain. And we showed that the interventional studies using symbiotic diets only affected the intestinal and the systemic immune phenotypes in the mouse models we tested here, except for the valproic acid model. So this beautiful 7-year journey, this was the dream team in relation to the preclinical studies. So we had the people from INRAE, Lea Rossini, who is now a doctor working at Cork University College Cork in the group of John Cryan, but also Sylvie Rabot and Laurent Nadon were exceptionally good collaborators in our preclinical studies. The people from Danone, Johannes Kostopoulis, Himanshu Kumar and Guus Roesler. My team in Utrecht, Naika Prens and Lucia Peralta Marzal, who did get their PhD degree, and Yangpeng Zheng, under the guidance of Paula Perez, who's still in Utrecht. Naika is now in Lausanne at the University of Lausanne. Lucia is currently looking for a nice postdoc position, and Yangpeng Zheng went back to China. And then we had a beautiful collaboration with the people of Tampere University in Finland, Carolina Salenius and Raya Autio and Jake Lin, who really helped us to digest the massive amount of data we generated. And this all started with the support of the people here in Salerno, the people from ASL Salerno, who really helped us to recruit the families and get the stool samples. And a big, big thank you to all the children and families who participated in our studies. Thank you very much.
Thank you, Aletta. Thank you for your impressive data. We have the opportunity to ask questions at the end of the presentation of this morning. And now is the turn of Dario Siniscalco from University of Campania, Luigi Vanvitelli. He will talk about epidemiology and risk factors.
Thank you, Massimo. Good morning, all of you. Thanks for coming and listening to me. So I'm going to talk with you about the observational arm of the GEMMA project and what we achieve in terms of autism prevalence in our GEMMA cohort of the families requited. So I don't want to stress or to teach you about autism, but just for the people that are facing for the first time with autism. You know that it was described for the first time in 1943, here the publication by Leo Kanner on Pathology Journal, and what we define now is a lifelong neurodevelopmental disability that usually, or at least, before starting GEMMA project, or at least in Italy, it's diagnosticated about 3 years of life. Is it too late? Maybe for an early intervention or for early diagnosis, 3 years of life is too late. And it is one of the goals of the GEMMA project. It's a neurological disorder, autism, that affects the area of social interaction, of communication skills, and some behavioral issue. The World Health Organization classifies autism as a disease in the disease list, but now we are talking about a disorder, not a single disease. The autism spectrum. So weakness, social interaction weakness, decrease in social communication, increase in repetitive behaviors, in sensory use, and also in self-injuries issue, above all for the teenagers. But what is missing about you? So, what is missing in your opinion? It's missing a biological marker. Indeed, we perform early diagnosis or late diagnosis without a biological marker, only by the observation of symptoms. It's for me, I'm a chemist, it's very hard to think about this a disease, a disorder without a biological marker. Second goal, one of the goals of the GEMMA project. So nowadays, according with the global burnout disease just 1 year ago, we have worldwide, as a number of the people with autism, more than 50 million worldwide, about more or less the whole population of Italy. Can you imagine this situation? Of course, males are about 4 times of the females, and we could talk about the genetic predisposition in males rather than females. And then, just some years ago, 2019, before GEMMA project, still according with the Global Burnout Disease and the University of Oxford, in the 3 main countries that participated in autism recruitment for the GEMMA, US, Ireland, and Italy, you can see that it's about the same autistic percentage, about 0.7 in respect of all population. And here we considering all the spectrum. So about the classical autism, that the classical autism is divided in 3 main subtypes: mild, moderate to severe, and severe. And remember, usually the severe classical autistic children do not speak, have a very, very hard problem in language. But it's a spectrum, so from classical autism until to Asperger's syndrome or high-functioning autism. So in each country that participated in GEMMA project, in US, about age 8 years, Alessio told before that we faced a dramatic increase, exponential increase of autism prevalence starting from 2006, according with the CDC, Atlanta. We had 1 child with autism on 110. Until now, the last data, 1 autistic child on 31. And if this very important association in US claimed that in only 20 years, from 2000 to 2020, we had an increase of 370% autistic people. But now, we have almost 400% in US. You know about a similar disease? I don't know. What happened here? When the curve changed the positive phase. In my opinion, it's not only a genetic disease. You can explain this progression, unbelievable progression, with a single gene, inevitable single gene. No. So Alessio told before about a genetic predisposition. Yeah, it's true. You know that a genetic predisposition, but we are talking about de novo mutation, not inevitable mutation. So hi Marco. The parents could also not have this genetic mutation, but genetic predisposition, de novo mutation, single nucleotide polymorphism, and above all, epigenetic variation. It's because maybe genetic genes are not inevitable in autism, but what is inevitable is the environment, because the environment where the family could affect the infants, the newborn. And the environment affects autism. How? Genetic difference in epigenetic change. So how the genes change their expression without affect mutation, without affect DNA sequence. So now we have about more than 32,000 autistic children with 8 years in US on 1 million of peers, of age-related children. It means 3.2% of autism prevalence. Remember this data because we achieved some different results in GEMMA cohort. In Italy, the last data was some years ago, 2019, from Istituto Superiore di Sanità and Health Ministry, Ministero della Salute. They told that in the age between 7 to 9, we have one autistic child on 77. It means that we have at least 13,000 autistic children, 1 million, and it means 1.3%. In Ireland, also in Ireland, autism prevalence shows strong upward trends. Interesting, exceeding the European and Italian average. Is the environment different? Indeed, according with the Department of Education, now we have one autistic child on 20 children, is 5% in each school-based population, and this is a threefold, 3 times increase in less than 10 years. So, National Council for Special Education reports a prevalence of 1 on 65 among all the student population, not only the young, and about the adult, we have 1.2% over 18, but maybe is an underestimation because it could be another 4.5% that without an official diagnosis of autism. So, when we thinking about GEMMA project, one of the main problem was that, a family that already has an autistic child is very difficult to think about a second or third birth, another child, no? Indeed, already some years ago, JAMA Psychiatry reported that when in a family you already have an autistic child, there is a strong decrease on the probability to have about a child, to have about another child. Because, the stress, the family burden that affects the family with just one autistic child. One of the parents, usually the mother, has to lose the job to take care of the child. This afternoon, we could talk about the political, the societal regulation and the situation here, at least here in Italy. So our clinical design was, we required a family with autistic child and with a newborn, an infant, in 6 months of age, or if the mother is pregnant. We collected the biosamples as blood, and we characterized a new protocol to extract peripheral blood mononuclear cells. I love these cells because they are the cells that my target for my autistic research. So we decrease the volume of blood to decrease the stress for autistic child. And we collect blood, stool, urine, and saliva every 6 months until age 3 years, just one time by the parents and from the siblings, already autistic siblings. And then we performed several behavioral assessments, behavioral tools, each 6 months until age 3, but the first assessment was due at 1 year of life. It's very important. And some of these assessments were observational with both apparent interview and so on, or else too. And also we collected biodata, dietary, medical, behavioral, and environmental data in REDCap database. So our results. We enrolled in our 3 different centers, Ireland, US, and Italy, 344 families. And we had 74 new infants diagnosticated with autism. We can see the difference in the three countries. In the US and Italy, we have the same number. We cited 74, 64, and 63 families with a prevalence of autism newly diagnosticated 4 new infants, while the Ireland recruited 217 families with an unbelievable number of 66 newly diagnosticated children with autism. So, it means that we have in GEMMA cohort 21.5% autism prevalence. I bored you before with the data in US, in Italy, and so on, in Ireland, just to show you that our autistic prevalence is much more higher than the official data of the single countries. And we have 6% autistic prevalence both in Italy and the US, whereas 30% in Ireland. In the left part, you can see that the average age and the range of the enrolled children. Then, 49 children, newly diagnosticated autistic children, were enrolled in interventional harm. Geraldine, this late morning, will talk about our results. And just let me thank a family here that I know is here, a family, the Italian family that was enrolled in interventional arm with the symbiotic treatment, and they experienced very good, very nice results. Then we used as a new tool for the behavioral assessment the BOSA, the brief observation symptoms with autism. It's remote. You know, you can, it allows an early detection for infant at risk, rapid, of course, You can observe the possible autistic child remote by telehealth, and it's available as a standardized tool. We performed more than 500 BOSA with online telehealth assessments in all the project, considering both interventional and observational trial. And for Italy, let me thank, it's a final meeting, we have to thank, let me thank Rosario Savino that is here, Veronica Sferragatta still here, and also Nicola Antonucci and Domenico Boll, for not only for helping us in recruitment tool, but also for evaluation and assessment. Taken with BOSA, we had the age at the first positive BOSA was 12 months. Next, as our protocols, we need 2 positive BOSAs or another external diagnosis with another tool. In example, in Italy, official tool is EDOS2 to claim autistic child. And the age of diagnosis was as a peak of the Gaussian curve at 80 months, you can see there, and as a second peak between 24 and 25 months. I think this is a very important first result of the GEMMA project, because you remember my first boring slides about Leo Kanner, and I told you that we, before GEMMA project, we diagnosed the autistic child about 3 years of life. Now, with the GEMMA project, we can perform, we can claim autistic diagnosis between 18 and 24 months. So in this way, we decrease the months, the year of the first diagnosis, or the definitive diagnosis of autism. So in this way, the child can undergo to early intervention. We recruited overall in the 3 recruitment centers, most by, you can see, about 60% by social media and web advertising, then from physician or medical referral, and from the company. As a company, we have an association, foundation. Thanks again to Enzo Abate for coming here. By the 3 countries, you can see that US recruited by most by company, association, and then by both by social media and medical record. Then here in Italy, we recruited most by our collaborator, medical physician, and then by association and foundation. Whereas in Ireland, maybe it could be a first difference, a first change. And Ireland has a national center, very new national center for autism. They recruited by social media, most, about 19% by social media and web advertising. So after me, Thanos maybe, we'll talk about the omics integration, how we can fit all the biological information with the autism prevalence. And I have just to thank again Antonietta Cicalese for the strong, helpful, we can work 24 hours, 7 to 7. Also Anna Schmitz, and let me dedicate this my entire project to the memory of my father that passed away just 2 weeks ago. Thank you for your attention.
Thank you, Dario. Thank you very much. Now we have, the last session speech of the morning, the first part of the morning, Thanos Biopoulos and François Iris, or Thanos alone, maybe.
Hello, everybody, and thank you very much for inviting us in this beautiful meeting. I'm here to present our results from behalf of Biomodeling Systems. So we are the partner that does the biological modeling. We will try to explain to you our view on how autism is developed in utero and how the omics data that we got from the program seems to corroborate our model. So a key contributor to this project was Francois Iris, which due to a last-minute inconvenience couldn't make it, but he sends all his greetings and salutations. So, the down one, okay. So our first objective in the project was to build a scientifically robust model in order to explain the in utero ASD pathogenesis, the emergence of comorbidities, and most notably the GI symptomatology, while also the complexity of ASD genetics that all the previous speakers talked about, which is actually that we have multiple gene combinations that could give rise to autistic behavior. However, these are not always causal, and that we can see between siblings that are autistic, that they only share around 30% of the genes. And also this variability that you can see in autism, because if a gene would have a neurodevelopmental effect, it should also have somewhere along the line a somatic effect. Well, this we don't see in autism. So it's more than genetics. So in order to do the modeling, we started from a structure modeling approach, and we were based on well-established neuroanatomical features. So what we did is that we integrated what is known in literature, databases and everything, on which parts of the brain are really affected, why there is altered connectivity, or why some regions of the brain are overdeveloped compared to others. And we try to link that with the symptomatology of autistic behavior. So while doing that, we tried to explore three different aspects all the way along the model building: the parental origins, so the role of gestational environment in ASD, the role of maternal inflammation and also gut dysbiosis in the mother, also the brain-gut link that Alessio very well talked before. So how brain connectivity is linked with this dysbiosis and how it's supported. And also the mechanism, so what drives broad gene expression changes that shape the autistic brain. So we produced the first complete neurodevelopmental model for ASD, which with identification of causal mechanisms and correlations with symptoms, while we could narrow down causality to a slim developmental window around the gestational week 6 to 12 or 6 to 20. And according to this model, we knew exactly where, what to look in the omics result to see if we can corroborate our model or not. So, due to the limit of time, I will try to give you a brief overview of the model. The thing is that the brain structures that are most affected in autism are the prefrontal cortex, the anterior cingulate cortex, the amygdala, the temporal cortex. And these regions seem to have an overdevelopment during the first years of life. And then we can see a reduction of neurons in the adult time of these people. So, and also they express altered connectivity inside the brain. What we found out is that these regions are spatiotemporally linked during early development. So during early development, we have the neural tube that's being developed, and there, all these regions are tightly linked in a structure called the pallium. And during this period, this is the most important part, is where the brain blueprint develops. This means that you will have some immune cells that will start forming a grid, and this grid is what will drive all future neurodevelopment. It's like the blueprint for brain development. And this is what will also drive neurogenesis, migration, and differentiation of the neurons. How this is done, these initial cells, which are called radial cells, they get their definite positions by following some cues in neurodevelopment. There are many of these cues, but one of these is serotonin 5-HT. And by following the serotonin cues, they get to the final positions and they drive neural development. They do the initial grid, and then neural development follows these first cells. Arborization, so connectivity, also depends from 5-HT. But I will explain a little bit of that in the next slide. So what we also see, what we know, is that maternal inflammation produces serotonin. How is this done? Because the immune reaction produces tryptophan. Tryptophan can be converted to serotonin, while also the gut of the mother produces serotonin during inflammatory conditions. The thing is that during the developmental window that I was talking about, so gestational week 6 to 12 or 6 to 20, the placenta allows the surplus of serotonin produced in the mother to reach the embryo, which this will change the neurodevelopmental cues for the initial brain blueprint to be built, and might have consequences in brain development. So if we want to see this in a bit more detail, here we can see the maternal side and the fetal side, and during these weeks, gestational week 6-20, if we have serotonin crossing the placenta, what will actually happen is that serotonin will alter the initial neural migration, placement, and arborization of neurons. So you can see that the neurons will not be in the place that they should be, or we're talking about a spectrum, so there is not a place that should be, it's a different development. And of course, and what else we will have is that serotonin will, in combination with another hormone, reelin, will dictate how the arborization will be made, so how these neurons will be connected. We also have inflammatory mediators from the inflammation of the mother that could cross the placenta and reach the embryonic environment. And this is the most important part, I think, because this explains the genetic variability of autism. So these inflammatory mediators may produce an abrupt change in the fetal environment. And this can induce what we call the epitranscriptomic changes. It's not epigenetic, it's epitranscriptomic. So abrupt changes in the environment can start a mechanism that will only change the gene product and not the gene itself. So it changes the RNA. There are 3 ways to change it. So we can have a substitution to adenosine to inosine that will change the coding sequence of the protein then. We can have a poly-A adenylation, which will change the stability of the messenger RNA and therefore the end product. And also we can have the creation of microRNAs which change the expression of the RNA molecule. All of these 3 will have consequences in the final product of the protein. And these consequences will be derived in a very narrow time window, the window of the environmental change or the window of the inflammation, and they will have a specific driven expression because because they're going to affect only the genes that are expressed at this exact point. So this could only influence brain development and not later somatic development. And as we've seen, there are many synaptic defining proteins that are very prone to these epitranscriptomic modifications. So these A2I, this epitranscriptomic modulation will lead to anatomical and neuronal network connectivity alteration that will affect CNS, central nervous system, for life. So here we can see that it's not the genetics, the highly complex genetics that are responsible for ASD. it is the gene themselves, it isn't the genes themselves that are affected by their transcripts following site tissue-specific editing. And this is why, although clearly involved, the plethora of strong autism candidate genes do not have the somatic effects expected. So in this slide, I will try to wrap it up visually. So what I'm talking about all this time, that if there is a cross of serotonin and inflammatory mediators during a very short developmental window from the maternal environment to the fetal environment, both mechanisms will alter the initial brain blueprint that will guide all later development, leading to a wide range of outcome and affectations. You can see the grid there, how it could be modified, and this is what will form all future connections. So that would explain the vertical column disorganization that we see in autism, the excess of neurons in the start, the altered excitation to inhibition ratio, and of course, altered connectivity at the start will change synaptic pruning, as very well Alessio said before, and this will also change the signal to noise discrimination ratio, and we will have a spectrum of degrees of affectation, which is the spectrum of autistic disorders. So this model confers the observed gene randomness and clinical heterogeneities encountered in ASD and ties up with maternal inflammatory reactions, giving us a short inflammatory window from the mother that could influence brain development. So what are the systemic consequences of a model like this? As we well said, we saw that very often the autonomic nervous system shows a more active stress response in children with ASD. What we can see here is that the autonomic nervous system has two different branches, that one balances the other. So we have the parasympathetic nervous system, which is what we call the rest and digest system, and also the sympathetic nervous system, which is the fight or flight system, so the active stress system. As you can see, the parasympathetic nervous system stems for the limbic structures of the brain, which are the more affected during the developmental window that I was talking about, while the CNS stems for the thoracic vertebrate system, which is not affected during the developmental window that I was talking to you before. So it's not an overactive sympathetic system, but it's more a parasympathetic system deficit that shows this active stress response in autistic children. So what's the link with the gut? I think it was pretty obvious from the previous talks, but the brain is connected to the gut's immunity through the vagus nerve. The vagus nerve is a parasympathetic system component. So what the parasympathetic system does is that controls the cells, Paneth cells, goblet cells, that are responsible for the gut's immunity. So if the parasympathetic system has a deficit, then it will allow dysbiosis in the system because it won't resolve or it won't respond to the signal from the enteric system immediately, allowing a dysbiosis to be built. At the same time, the parasympathetic nervous system, through the circadian rhythms, is connected to the liver. And the liver itself also modulates digestion. And there are microorganisms that can feed on digestive system, so they can feed on bile acids, etc. So if the immune system is late to respond, what will happen is that we will have an unresolved and persistent dysbiosis. And this will lead to systemic inflammation that we will have effect and in the gut and in the liver, creating a vicious cycle that is very difficult to resolve. And when we had this model, the omics start coming from the system, from all the children that we recruited, from all these branches of the clinical studies. And what we see is that, indeed, inflammatory signals, systemic inflammatory signals, increase after age 2, which age 2 is the moment where the immune system of the children reaches maturity. And what you can see here, it's IL-1B and IL-6 that are rising from 2 years onwards. Anti-inflammatory signals, the brakes, as we say, the brakes from the parasympathetic system are clearly non-existent, so very down. IL-10 is the brakes. We don't see. IL-10 is downregulated compared to normal developmental children, which means that the parasympathetic system can control the immune system of the gut. We see that is a chronic condition of enteric origins due to the increased antibodies, enteric antibodies, IgA, that we see on these autistic children, while also we see that it's a chronic condition because the acute markers of inflammation, like interleukin-8, are downregulated in these children. We further see dysbiotic microbial signatures. As I said before, if there is a loss of the immune control of the enteric system, and there is also an overproduction of bile acids from the liver, out of sync, let's say, not overproduction in the bile, that will favor the organisms that can feed on that. So we can see clearly from the microbial signature, from metagenomics, that we'll have an accumulation of bile-eating bacteria, mucin-eating bacteria. Mucin is the protective layer of the gut. And also, we can see also glycan-feeding microorganisms, which feed on the glycosylation features of the enteric tube. We also see a decreased enolase level. Enolase is oxytocin mimicking hormone produced by bacteria, which has soothing effects in the brain. And this was confirmed by the fecal metabolomic signatures, where we saw the increased bile, mucin, glycan-derived metabolites, confirming the dysbiotic profile that we saw from metagenomics. And, as Alessio said, we also see nonulin that confirms that there is gut leak, and this gut leak might produce toxins going to the circulation and then feeding that vicious cycle and maintaining chronic inflammation. And the last results that came, I think, yesterday, were that we can see reduced cholesterol moieties and apolipoprotein moieties, which are also connected with the out-of-sync release of bile acids from the liver. All of these results seem to corroborate the data and support the autistic pathogenetic model, which is points towards a neurodevelopmental, chronic, low-grade, autonomic nervous system, gut-sustained inflammatory state with different degrees of affectation that are due to the initial blood formation. So, in summary, taken globally, the mechanism that we revealed by this model can explain the in utero neuroanatomical origins of ASD, the neurofunctional characteristics of ASD, the ensuing autonomic nervous system functional abnormalities, the origins of the ASD-associated dysbiosis, the reason for its persistence, the apparently high complexity of the ASD genes, the global enormous clinical heterogeneity encountered, while also suggesting mode of symptomatic treatments and producing biomarkers for early diagnosis. What are the perspectives now? I want to stress here that it's very important that we can still continue and pick up to all these cohorts that we have created during the GEMMA project, because there are invaluable data that could give us more profound knowledge about what autism is and how to affect the lives of children, their parents, and adult people with autism. So the perspective is to respond to the previous speaker, that we need to find a valid biomarker. And these valid biomarkers could be the ratio of the metabolites produced by the autonomic system. So the PNS, the parasympathetic to the sympathetic nervous system. This could give us an accurate early time diagnostic protocol based on biological parameters. Further on, now we have all this data produced, we can pick if it's not only epitranscriptomics, but they also can be epigenetic modifications. So we know exactly where to go and go back to the data and see if there are epigenetic regions that affect whole expression levels of systems. Then we can start thinking on ameliorating symptomatic treatments, not only by giving pre and probiotics, but also trying to modulate the PNS activity as for the intervention not to be transient, but to be long-term. Because if we can stimulate the parasympathetic nervous system, it will control the immune system of the gut, and then the interventional with pre and probiotics treatments will be more efficient. And this also stands for the fecal transplantation techniques that exist up to today. And then we can start thinking about, it's very early, but we can start thinking about maternal ASD preventive strategies if there's something to be prevented in maternal inflammatory and dysbiotic biomarkers. And of course, if such a thing that a syndromic ASD exists, because I don't believe it, because if syndromic ASD existed, it will be only related to genes, and they will have consequences, somatic consequences. So this could be another form of autistic-like behavior that cannot be described as being inside the spectrum. But also, for these kind of pathologies, we could start having parental gene assessment, as we now know where to look in the data and see if there are modifications. So with that, I want to thank you very much for having us here. And thank you.
Thank you, Thanos. We have 15 minutes for a coffee break, so see you back at 12:15.